Good morning, Professor Feng, colleagues, ladies and gentlemen. Welcome to this pair distinguished lecture. You probably know that we hold this lecture series for quite a few years, and we always invite the top-notch scientists and university administrators to give a lecture. So today, I will just invite our Associate Vice President, Professor Wang Zhongkai, to introduce our speaker. So, Professor Wang, please.
Okay, good morning, ladies and gentlemen. It's my great pleasure to introduce our distinguished speaker, Professor FENG Xiqiao, a Changjiang professor in the Department of Engineering Mechanics in Tsinghua University. Professor Feng is also academician of Chinese Academy of Science. I think, when we introduce our speaker or when we talk about somebody in private, we always start with brilliance, achievement, and success. But sometimes we end with however. But for Professor Feng, there's no however. I have known him over 1 decade. He is always truthful, gracious, and knowledgeable. And Professor Feng bears his surname, Feng. I think in Chinese culture, Feng means jumping like a horse at high speed with momentum and mechanics. I think that seamlessly align with Professor Feng's research area, mechanics. By training, he is expert in solid mechanics, and later on, he spent his research to biomechanics. I think over the past decade, he already done great work, especially, he extended new idea, new model to explain new phenomena in cancer metastasis and develop a new method, even for drug delivery. And his last second given name is Qiao. In English, it's a bridge. I think Professor Feng is really a bridge to his student. He's always nice. He gives lots of effort to his students, especially advance their career. For that, and also for his outstanding contribution, he was awarded the title of, I think, Beijing Mingshi, the English name. Let's look at. I think he was awarded the title of Beijing Municipal Teaching Master. And beyond that, Professor Feng's scientific excellence has been recognized by many prestigious awards, including the title of a National Outstanding Scientific and Teaching Worker, 是中国科技工作者, I think, worker. Okay, not good translation. Okay, and also the National Natural Science Award. Without further ado, ladies and gentlemen, please join me in welcoming Professor Feng to give the distinguished lecture. Thank you.
Yeah, Professor Chen, Professor Wang, and Professor Zhang, thank you very much for inviting me to here and give the nice introduction. As Professor Wang said, my background, in fact, is fluid mechanics and solid mechanics. And I moved to biomechanics about 20 years ago. And I founded the Institute of Biomechanics and Medical Engineering in 2006. It is 20 years ago. And today I would like to briefly introduce my recent work in this field. In my institute, we would like to investigate through the integration of mechanics, biological sciences, and medical engineering, the fundamental principles in the life processes at different land scales, and to establish theoretical and numerical methods and apply them in medical engineering. In fact, biomechanics has a very long history. more than 2,500 years ago, Aristotle wrote the first book on biomechanics, that is on the motion of animals. He used the classical mechanics principles, for example, the level principles and the balance of forces and moments to understand the motion of animals. Later, Da Vinci used the same methodologies to understand the mechanics of life. For example, is the flying mechanics of birds. In the 17th centuries, Borelli wrote the book almost have the same titles is on the movements of animals. They developed systematics, dynamics of the bones, muscles, hearts, and lungs, and so on. In the last century, a large number of scientists study the mechanics of our bodies. For example, the micromechanics of our muscles and the micro circulations and also the hemodynamics. And here and several others is when the Nobel Prizes is due to their contribution to biomechanics, in fact. And about 50 years ago, Professor Feng introduced the stress-growth relationship. This is an important contribution. In the classical and in the traditional mechanics of biological tissues, normally we start the mechanical behaviors of a tissue, for example, bones, muscles, and make a test to stress the stress relationships or the constituent relationships of these tissues, including the membranes and so on. And the contributions of Professor Feng, Yuanzheng Feng, is that he said that the growth, degradation, and the mechanical behaviors of the tissues is greatly dependent on the stress of in the tissues. But his theories is still have some limitations. First, it is a qualitative curve with a large number of fitting parameters, 7 parameters. Also is limited to macroscopic tissues and is still lack of considerations of micromechanisms in the tissues. But this is still the foundation of modern mechanics. This is important. And what we would like to do is that we would like to consider the mechanical, chemical, and biological coupled systems of the tissues of cells and physiological and pathological processes, for example, in the different organs. In the field of biomechanics, we have a large number of very important key issues. For example, the fundamental principles of biological tissues, which are greatly different from non-life of engineering materials and structures. For example, what are the statistical mechanics of the molecules and cells in our tissues? I would like to discuss this in a moment. And also the basic principles of the cell sensing, activation, transductions of cells and molecules in bodies, and also the principles of mass and energy transport in our bodies. And for example, if we consider the cells, these are several cells on a substrate. The cells have very long distances. For example, I know Professor Wang needs anything, and then I give a nanotube. There's a nanotube between the 2 cells and make an efficient transport between 2 cells. And they have a network between the cells. And a large number of mechanical problems also exist in our bodies. For example, we need to measure the mechanical properties of different tissues. And also we have a large number of instability phenomena and fracture behaviors, and also the microstructure evolutions in the tissues and different constituencies. And also we have multi-field coupling mechanics problems, and also the multiscale and problems. Also, we are interested in the morphomechanics of different tissues in the body. For example, how the network, your bodies are formed in these forms, in these very elegant forms. And in this talk, I would like to talk the dynamics of cells at different length scales. The cells in our bodies have more than 300 types, which greatly different in the structures, in the dynamic behaviors, and we would like to study them. How we can consider these issues is, first, we consider the Newton's law. The force in the cells are very complicated because the forces in the cells are very difficult to measure, and they are coupled for the different mechanisms, and they have active forces and passive forces, and which are very difficult to be qualified. Another one is the constitutional structures of the cells, and they are greatly different in the structures. For example, the neural cells and other cells, and they have greatly different constitutes. And another the dynamic behaviors of cells is they deform, they move, they adhere, they spread, polarize, oscillate, and a large number of different dynamic behaviors. They are coupled to each other in the body. And would like to discuss these dynamic behaviors at different length scales from the molecular scales. For example, the interaction between the medicine and the proteins, and also the organelles, the cell skeleton, the cell membranes, cell nucleus, the dynamic behaviors, and the division and migration of cells, and also we move to the organs is brain, heart, lungs, and the systems. Now another key issue is that how the mechanical, chemical, and biological mechanics are coupled to each other. For example, if we consider such as cells and the external stimuli, in the case there's external forces is acting as cells, and they will introduce a large number of the pathways, biological pathways. And for example, they introduce the actin polarizations, actin stabilizations, and myosin activities. And these are the findings is a biological scientist. In our study, we would like to transform these biological findings into mathematical equations, and then transform them into the mechanics theory. For example, if we consider such the pattern on the cells, we can find such a pattern patterns on the cells, and how we can understand these things. First, these cells are subjected to stresses between different cells, the neighboring cells, and the stresses or the stretchings is introduce the deformation and the conformation changes of molecules. And these are the force sensing and activations of corresponding pathways. And then the cytoskeleton, membrane, nucleus, they will deform and respond to stimuli. And then they will introduce the mechanical and functional changes of cells, which are adaptive to the external stimulus. And finally, we would like to transform them into a series, and they propagate in the systems. The next issue is how we can determine the forces in the studies we are in the system we are studying. For example, if we consider such a embryo, this is a cell embryo. In the embryos, there is a tail. There is a tail. The tail is banded. Is banded. According to the classical mechanics, if the beam is banded, there should be a force subjected to the beam, to the tail. And normally, this said that this bending was caused by the shell. For example, the egg, that shell. During the development of the embryos, the shell acting forces on the tail, so the tail is bended. Is it true or not? We don't know. Let's consider it. And how we can consider it? We remove the cell during development of the embryo. We found that the tail still bent. There's no force acting on the tail. What happened? This happens that the polarization of the tail on the frontier and the back layer of the tail, the cells division have a different speed, so that the internal forces causes the bending of the tail. This is not an external force. We must measure these forces. Another example is that we consider a stem cell in colony. And if we consider a droplet, a water droplet, like Professor Zhang Kaiwang, that the surface tension is on the water surface is this shape. The droplet have such a shape. The same shape happened in the stem cell colony. Is there a surface tension in such systems? Okay, let's determine it. And we measure the shape of the cell nucleus in the tissue. On the surface, in the interior of the colony, the cell nucleus has spherical shape, but on the surfaces, they have a oblique shape, like a pancake. This means that the different shapes of the cell nucleus indicates they have different stress state. On the surfaces, there should be a surface tension in the colony. And what is the where is the force? Who generated the forces? Okay, let's show it. And we make imaging of the cell, of the actin filament, myosin, and other moleculars. And we found that on the surfaces of the colony, the cells automatically generated a surface tension like a cap. This causes surface tension in the colony. But this cannot be seen in the normal cases. Now let's move to the second section. This is the rankling or instability phenomena in our bodies. In our bodies is there greater different patterns on cells. For example, the complicated the shapes of tumors and the rankles on the body. We studied these things for more than 20 years, and we have a book on the surface rankling mechanics. But in our bodies, we have different type of the instabilities, including the ulnar buckling, the Turing instabilities, and the biomechanical instabilities. What are their differences? The first one is ulnar instabilities. These are totally the same as in the classical solid mechanics. They transform between the bending and tension and the compression energies. The Turing buckling is totally different, determined by the chemical energies. But in these cases, they cannot be explained by the classical Turing instabilities or the ulnar instabilities. We should consider the coupling between mechanics, chemical, and the biological cues. If we consider the instability phenomena, we should study from the following four aspects of the mechanisms. The first one is energetic mechanisms. And the second one is the mathematical equations of the governing equation. The ulnar buckling is determined by elliptical shape, so they cause the ulnar instabilities. And the third one is the temporal features, which including the speed the frequency, the propagation, and the evolution. I will give examples showing these things. And the last one is the special features of the pattern, of the Rankine pattern or the chemical pattern thesis. We consider them from the periodicities, symmetries, polarities, chiralities, and temporal spatial coupling with evolutions and the correlation between the local and global patterns. I will explain this in a moment. And in fact, we study the instability phenomena from different angles, from the actin filaments, cell skeletons, the cells, and the collective cells, and so on. Now let's give 3 examples. The first one is a single cell, the second one is collective cell, and the last one is the tissue. We can only give several examples. First one is this wave propagation in single cells. And you can find that they have such a complicated pattern propagating in the surface of the cell. What's the reason? It's important that we have a clock on the corner. This is 1 hour and nearly 2 hours of the movie. This means what this means? This means that this is not elastic propagation. This is a chemical mechanisms. And because the elastic propagation have a very high speed, more than 100 meters per second. And this is not chemical propagation. The chemical propagation is also faster than this one. Okay, now let's consider this. This is a figure of the mechanisms. On the surface, there are cell membranes, and beneath is a cortex layers. When the cell is subject to external forces, they will introduce such a complicated mechanisms as I mentioned above. Now let's write these biological findings in mathematical equations in these senses. This is a mathematical formulation of the mechanical chemical coupled mechanisms. And then we introduce these biological findings into active cell model of the cortex. And then we can find that the cell will exhibit different bifurcation behaviors, including the Hopf bifurcation, the pitchfork bifurcation, and the oscillation and the Turing patterns and different constitutions. And this can explain many cell behaviors and different conditions. For example, the oscillations I will show you. A single cell can also oscillate in a different speed, which are important for the propagation and migration of cells. So for single cells have a spherical shape, they can accept such a different phase diagrams. In this case, it's stable. In this case, it's oscillation. And in other cases, it can have a complicated patterns and the wave propagation is like a chiral patterns. This is comparison of our theories and the experiments. For more details, you can find this paper in PNAS. The next example is the dynamic instabilities of collective cells. And this is the movie on such an embryo. This is the happened during the Drosophila during closure. And why such a pattern, such a wave propagation is on the tissues happened? This is important for development of the tissue. In fact, this is also simple. It is caused by the feedback of mechanical stretch, mechanical strain, and the chemical activities. For such a tissue, each cell is dynamics. They have a bifurcation, and they have a oscillation, they have a division, they have during development. And what's the result of this greatly different perturbations is? And we don't give the details of mathematical theories, I just give the results. And the first result is the stable tissue. For example, if you observe the skin, it is stable. If you give some perturbations, it's also stable. They don't have such dynamic behaviors. But other cases, they have except the Hopf verification. This is a subcritical verification in mathematics, and the tissues will change such a stable oscillation or limit circles in dynamic behaviors. And so this is the theoretical results, which is very similar to what happened, what we observed in the experiments. And the last example is the instabilities in the landscapes of our tissues and organs. For example, we cooperated with the hospital and made a real diseases. In China, we have very large populations. Many doctors, in fact, cannot judge what these photos means. It's caused by infection, cancer, or other diseases. And so we would like to make theoretical analysis using such AI techniques to give the information into the instrument. Once the instrument gives the results, they can help the doctors to determine, to judge diseases. And also this diseases, varicose veins, also happened in many aged pupils. This is especially for us sitting in the offices every day. So we need to travel. And also the instability also happened in the organ on chips and other 3D printing technologies in the tissues. Okay, now let's move to the next section, the dynamic behavior of the collective cells. In our bodies, in the tumors, in the embryos, and other tissues, which always involve the dynamics of a large number of cells. The cells collectively to achieve the functions. And in fact, we studied these things for more than 20 years. And Professor Bolli, Professor Yue Shao, and Professor Guanghui Xu, and this also study these phenomena, and they published a large number of papers. And so in the past years, we have established a platform to study the dynamic behaviors of collective cells and make the theoretical studies. In the last years, we initiated a major research plan of Natural Science Foundation of China. We proposed by this project with Professor Xuebiao Yao and together. And this project is about more than 200 million Chinese yuan. And in this, can we move this? This is a movie. And this is one type of cells. This is another one. This is the name of the cells. In the beginning of the observations, we have a wall between the two types of cells, and then we remove the wall and we observe the interaction of the two types of the cells in order to study the cancer propagation in our body. And this is the movie of the cell nucleus or the cell skeletons of the cancer cells. And this is another type of cells with the time. You can find that they have different shapes and different behaviors. And we measure the the velocity of each cells, the position, the shapes of each cells, which including more than 3,000 of cells. And we observe them in 2 days or 3 days. And then we measure we track individual cell pathways and the interfaces and all those things, and make a statistical analysis. From the imaging, we can calculate the stress fields and the strain fields in the tissues and analyze their results. And then we make a theoretical numerical simulations of the tissues. They have different dynamic behaviors. For example, we make such a large scale of simulations of the dynamic behavior of the cells on the surface of the curved systems. Now it's important that we would like to consider the statistics of the cells. In the classical physics, we have such 2 well-known distributions of the velocities. The first one is the Cosine distribution, and the second one is K-gamma distribution. They have different shapes. We want to know why the distribution works in the active cells. Now, let's compare the 2 functions with our measurement. We found that neither of them works for cells. This is Gaussian distributions. They are greatly different between our measurement. For the K-gamma distributions, you can find that they have greatly different with range of low velocities. What is the reason? The reason that the active cells, the molecules, involve the transport between mechanical energies and chemical energies. They have not been considered in the classical series of entropy. About 30 years ago, a Brazilian physicist, Thales, proposed a high-order theory of entropy. This is his definition of entropy. It's called Thales entropy. From this series, it can be reduced to the classical Boltzmann-Gibbs entropy. From this theory, we derived such a K-Gaussian distribution. This is different from classical physics. And now we compare this theory with our measurement of very large amount of entropy. They have very good agreement. Now let's don't give the details. Now let's move to the next section is multi-scale modeling of biological processes. In the last year, we studied another project of National Science Foundation of China, collaborated with Professor Lu Mingli. He is the president of Tsinghua University, and Professor Hua Jiangao, and so on. We studied the mechanical biology of brain diseases and neutral stimuli. As you know, in China, we have a large number of older people which have such a PD and AD diseases brain, which have a very high ratio than other countries. And Professor Lu Mingli, developed a deep brain stimuli technique to treat such diseases. And we studied the mechanisms, molecular mechanics, and make simulations. For the brain, it's very, very complicated. We need to consider, make a simulation from different aspects. The first one is the microstructure and the macrostructure scales. As you know, we have a neural network in our brains, but this is only one network. There are also many other networks, for example, the blood vessels and the network brains and other immunological systems. Another one is time, because the brains involve electrical field, mechanical deformation, and chemical diffusion, and so on. Another number is the topologies. The neural network is very complicated. This is image made by Google. It has only 1 kilo micrometers. And this is a photo of a single neuron. It has more than 10,000 of neural synapses. So if we want to simulate a brain, the dynamic brains, what is the date? We have about 1,000 billions of neurons, and they have such a large number of signal processes which functions as the signal transduction. And also they have a large number of other cells and the ECM and the liquid and so on. This data cannot be even recorded by our supercomputer, regardless of dynamic evolutions and diseases and the evolutions with diseases for many years. So we would like to use AI techniques to do this work. Currently, we have many famous AI models. The most famous one is AlphaFold. About 2 years ago, these 3 scientists won the Nobel Prizes due to their AlphaFold technique. But all these available AI models is still limited to the predictions of simple protein structures with a known long chain. This is make a function between the mapping from a long chain with static protein structures. It cannot consider dynamic behaviors of the proteins. The achievement of the proteins is at different length scales. So there is a great demand for numerical simulations in the study of cell dynamics and also the design of medicines. About 4 years ago, we are invited to write such a review papers to show how we should study the dynamic behaviors at length scales. So this review paper was finally published in CNS because we spent a long time to write this paper. And this is a framework we published 2 years ago in NECS Nano. And how we consider these things is with aid of AI. And then cooperated with other scientists. Okay. We established such a platform to understand the things. This is a local movie of the study. So now let's show the examples of our study. That is the cell migration is such a real tissues. And this is a real cancer cells in a 3-dimensional environment. The cancer cell needs to immigrate from one tissue to another tissue. It's a large number of greatly hard, soft of barriers, how it kind of works. We study these things. And this is our real simulation models. For example, if we consider the metastasis of cancer cells in such a tissue, For example, if we have two barriers, very hard barriers, and another hard barriers, there's very narrow spacings, how the cells propagate in such a narrow spacings. We study the mechanics of these problems. Okay, you can imagine that you are a cell. You don't have eyes. How we move, get out from this room? We use our hand to sense the environment. Okay, if we mess some things, mess up barriers, okay, we should increase the forces. But we don't know what is the environment. It is the wall, the door, or other structures. Okay. So we need to increase the forces. How can it increase the forces? So this process involves the mechanical senses and the mechanical transductions. They need to biochemical feedback to the forces and then make activation of the cell migration. These are really mechanical, chemical, biological coupled problems. How it can move. This is a real element which generates forces to sense and activate the forces, the movement of the cell. When this is a very thin element, filament. When the thin element is compressive, it will be buckled. So there is a configuration changes, and then this complex will compare at this position, and then it form another branches to increase the forces. So the number of the branches increase with the propagation and to increase the forces to move forward. But if the cells meet a wall, a concrete wall, it cannot pass through from these things. If they move such a tear, they can remove the tear. The cell can remove the tear by some structures. I don't tell you the details. So in this, when we meet a wall, we can open the wall. The cell can open the wall. When it meets the tear, they can move the tear. When it can meet the wall, the structures will become collapsed, and then they go another way. This is the first with the positive feedback. Another is chemical feedback, and also chemical reactions. Now let's give another examples of the series. This is the immunological system. And I just give one example is due to the limitation of time. We consider the lymph nodes. In your bodies, okay. In your bodies, we have 500 or 600 lymph nodes from the head to the feet. About 60 years ago, as you know, that we met the COVID 2019. And so we study the mechanics of immunological systems, including the lymph nodes. The lymph nodes swell greatly. For example, if I show this compression between normal lymph node and the swelled one, it can increase more than 20 or 30 times in volumes. More importantly, in the lymph node, you can find different types of the cells. They have highly dynamic behaviors. They work together, they react to each other to know what happened in your bodies and then make the responses. And so during these problems, the cells connect to each other. some cells move along the three-dimensional network, and the others is move in the liquid, in the spacing of the network, and they will respond to each other and make such a theory. The network still involve, they can expand, enlarge, and they can damage, broken. And this is the comparison between our theory and the experimental results. You can find that they have a good agreement. When in the beginning of the infections, inflammations is the lymph node swell, and then it reaches a stable state, and then it will be rehealed. And also, very interesting that the evolution of stresses in the tissues is it have 2 peaks. The first 2 or 3 days they have very high pressure in the lymph node, but then it will reduce to this level, and then it will increase again. And this is in first time of simulations. And this is a phase diagram of different pupils. For example, this is a person, immunological responses of a person who gets such diseases of, for example, cancer. The immunological system is very weak. And for this one is a young pupil. It's a very, very strong response to diseases, for example, to the COVID viruses. And we proposed mechanical measures for the immunological systems, and we use different parameters, but don't give the details. And finally, we defined a mechanical chemical coupled matrix to quantify our immunological systems, immunological memories. And this is the chemical parameters, biological cues, and mechanical properties to define the immunological things. And now let's give an example. This is the results of our series to understand why the COVID virus vaccines does not work well in your body. And the way to make the real simulations of the COVID virus vaccines is very weak. It cannot well work in your body. Now let's give the conclusion. In the recent years, we have studied the dynamic behaviors of cells and tissues. We would like to establish some theoretical and numerical methods for understanding the mechanical, chemical, and biological coupling mechanisms of these things and makes cooperation with hospitals. Now, finally, I would like to thank the co-workers, students, postdoctors, and hospitals. Now, this is my talk, and I would like to thank you very much for the attention.
Good morning, everyone. I'm Yuhua Tan from Department of Biomedical Engineering. So I have a great pleasure to moderate the Q&A session for Professor Feng's distinguished lecture. Because we have the audience in the room, and also we have the audience online. So for the audience in the room, so if you have questions, please raise your hand. And later on, for the online audience, if you have questions, please leave your questions in the chat box. I will grab those information later on to deliver to Professor Feng. May I start from the first one? So I guess majority of the audience may not necessarily working in the field of biomechanics. So Professor Feng, can you give us a little bit more information especially for those audience not in the field? So why people should care about biomechanical forces in the living system?
First, I think now it's a very important time. It's a good time to study biomechanics. First, in the past century, in the past 100 years, biological sciences have made very significant improvement, advances, especially from the cells, tissues, molecular systems. But most of these behaviors, the mechanical principles have not been known. No matter in the hospitals and in biological sciences, they have very high, very great demand for studies of mechanical principles. For example, why the protein can achieve the greatly rich functions. We would like to help them to understand these things. The second one is the development of mechanics itself. Solid mechanics, fluid mechanics have very complicated structures from the theory, numerical methods, and the experimental techniques. And these theories have been applied in aerospace engineering, civil engineering, energy, and many other fields. But we don't have such a systematic theories for biological problems, especially at the micro scales and the molecular scales, cell scales. For example, the cell skeleton, we don't have such a good theory. We should develop the mechanics itself. The third one is the major diseases in your body. In the world, there are tens of thousands of diseases which may happen in your body. And the forces play very significant roles in these diseases. In your brain, in the heart, in the lung, in the bone, in the muscle, and so on. Forces are always important. They capture, they shape the processes. But we don't have such a theory to simulate these things, because which are very, very complicated, as I shown in the slides. And in addition, in the recent years, there are many new fields in the biological sciences. For example, the organoids, organs on chips, 3D printing of tissues and organs, and so on. All these fields need mechanics. For example, if you want to make an organ or tissue by 3D printing techniques, you just imagine what functions we should achieve. But the results often differ from your expectation. What's the reason? The reason is that we have not understood the fundamental mechanical principles in the tissues. The things is different from your expectation. So we should study mechanics in all these things.
All right, thank you, Professor Feng, for the detailed clarification. So, yeah.
Thank you very much, Professor Feng. I'm Shujun Wang, the assistant professor in biomedical engineering. Actually, my research direction is AI and its applications in the medical field. And currently, the AI in virtual cell is very hot. I also noticed that you are working on the virtual cells. And currently, we are also doing the big data analysis on the single cell RNA data to analyze the cell state under some perturbations like the gene level or drug level, and to see how it will affect the cell. And I think it's your talk is very it's inspiring me about how to combining the like biomechanics, that kind of factors into the like the data science, that kind of I'm wondering, like I just want to seek your comments or your suggestions, like combining these two directions together.
Okay, thank you. This is very important questions. In fact, in the recent years, for example, in my university, Tsinghua University, a big platform is being constructed, which compare the professors from biological sciences, medical schools, and the mechanical and electrical fields. They compare the data from different areas, and we work together to study the problems in medicine, in diseases, in the basic biological sciences. So if you want to study the things, it's very good problems, very promising field of study. In the recent years, we have very large number of data, especially with the advances of high resolution of observations at the molecular stages and the other microscopic structures. So we should compare the different data together to make a real good simulation.
Okay, thank you.
Yeah.
Yeah, okay.
Thanks a lot, Professor Feng, for your distinguished lecture. And I work in the macro level of biomechanics, but I learned from your lecture that there is instability in the mechanical, chemical, biological environment in the human body or in the life. And to my personal understanding, the instability is not necessarily harmful. It may be beneficial to the life. So based on the technologies or research methods, the cutting edge technologies, do we have the way or approach to identify if the instability is harmful or beneficial? Do we have
Yeah.
Thank you.
In fact, this is really very difficult problem for us, for me to understand, for me to answer. The same factors is sometimes is harmful, but some other times is beneficial. They need to have a balance of different parameters, mechanical, chemical, and biological things. And in fact, it's also difficult to us to simulate these processes. And for example, if we use some vitamin, it is harmful or beneficial to our bodies. And we can simulate. And I have a student who is interested in these problems. And they can make a simulation. If you eat some vitamin, sometimes it is beneficial to our body, to the cells, but sometimes it's harmful. Yeah.
Hi, I'm Professor Almerker from BME. So I study a lot on the musculoskeletal system, and I'm truly inspired by your talk and the contributions that you've done to the field. I would just like to sort of seek your opinion as to what you think of the future directions of the field, particularly because from a musculoskeletal point of view, we study a lot of ECM rich tissues like tendon. And there's definitely a lot of interactions that go on between the cells and the tendon in terms of the biomechanics.
Yes, this is a very, very good question, but also very complicated to study. I don't have the real experience of study in this field. And in fact, our body are very complicated. They can change, they can evolve under different conditions. For example, in some diseases, the structures and the mechanical behaviors can change a lot. And we should, and I think we should make a comprehensive systems to integrate the evolution, the changes in different landscapes, different aspects, to make a real good simulations. And for example, if we consider the macro scale, for example, our muscle and bone systems is similar, I think, in the mechanical behaviors. And a robot with AI, a physical AI, can simulate these things, but it's very difficult to make a prediction because they don't know what the real mechanical behaviors of a patient. These patients large number of muscle cases, they have different mechanical behaviors, how it can make a prediction. If they cannot predict the dynamic behaviors, they cannot make a response. I think this is difficult.
Okay. So we have the online questions. Since it's live in Chinese, I'm trying to translate. So Professor Feng, in multiscale numerical simulations, how to integrate different like special tempo level of cellular behaviors into the same model, for example, from molecular to cellular level into a same model in the multiscale system?
Yes, in fact, this is still a challenging issue. It's parity for the simulations of the time scales. For example, in the your brain, the electric field propagates very, very fast, but the simulation is much slower, and the chemical reactions is much slower, and the diseases may last maybe 10 years or 20 years. Right. It is impossible at this moment to make a real couple of these mechanisms, and we must consider a simplified method. For example, if we consider the interaction of the medicine and the neurons, we just consider that problems, and we give these results to the next time and the next skills, and then to give the next one.
Right. Yeah. So we should simplify the system first. Otherwise, it's impossible. Yeah.
Not the concurrent simulation.
Yeah. So the second question from the online audience, that is, when you are dealing with those using the numerical simulation to deal with those very complicated mechanical response from the cells, what kind of challenge you face when you are using the numerical simulation in terms of accuracy and efficiency?
Yeah, in fact, I just answer this question. I have a student, Xindong Chen. He's studying this problems, and he opened a company, in fact. And for example, we want to see the design of medicine. We just consider the small molecules of medicines to interact with the protein, And at this moment, for these problems, we just consider the interaction of the different proteins and the medicines to make the results to show whether this medicine is good or not. It will probably work or not. If it does not work, we can forget this medicine. But for the other property, for example, you can see the migration of one cell through a three-dimensional problem. We can see the cells is in action with the surrounding species. They have the chemical reaction species. The protosome and other structures can remove the materials. If here that there are chairs, the cells can move these chairs and then pass through from these things. And we just consider the mesoscale, in fact. So this is a challenge. It's impossible to us to consider the whole structures from the molecular to the tissue. And for example, if we studied the behaviors of our brains or movement, how it controls our tissue species, our organ species, we must have simulations, the simplification species. And as you know, in the last year, the paper says that it does simulate how a fly uses his brain to control his
behavior,
dynamic behaviors. It have only, I think, about 300,000 of neurons. But in our bodies, we have much more, 100 billion. It is possible to simulate.
Okay, thank you. Yeah, the question in the back.
Thank you. Hi, Professor Feng. I think your speech is very it's very impressive. And especially just now you mentioned that a lot of dimensions from your speech, like the single cells, like the molecular proteins, cellular populations and tissue organs. And I observed that each of them actually owns their own independent structure. And it's very rigorous. But I have a question. I mean, if they don't share a common architecture, it means that maybe they cannot couple with each other. And especially when we run into some special environment like the diseases tissue, then how to observe it, how to distinguish which parts go to some wrong way. So I'm just curious about this question. Thank you.
Yes, because the problem, in fact, is very complicated. And we have a framework for study these problems. And because the cells, we have about 300 types of the cells. And we have a software to distinguish different types of cells and different of the ECM structures. And we have a basic framework. For example, most of the cells have the cell membranes, cell skeletons, cell nucleuses, and ECM have some common structures. And for the specific problems, we add the subroutine to determine whether it have the specific problems you mentioned.
That's very clear. May I ask another question?
Yeah.
I mean, from your speech, I noticed that you have mentioned that AI cannot predict anything because it's still restricted into some knowledge we have known, right? So when you mentioned the software things, do you think maybe if we want to discover some methodology to solve the problem I mentioned before, maybe we need to focus more on the propagation inside the AI algorithm. So maybe AI just serve as a accelerate tools to act it, but inside it we actually share the same thought as a cell experiment to focus on the propagation, maybe the longitude propagation and the cross-dimensional calibration to make it be more dynamic to observe it or supervise it. So maybe it will be more dynamic. May I understanding this way?
Okay. AI is a very important and powerful technique for us to study the problems of cells, molecules, and tissues. And everyone can use AI techniques. But many pupils don't know the details of forces in the structures. They don't know how to calculate the exact interactions of the medicine, molecules, proteins, DNA, and so on. While the available platforms in the network, AlphaFold, Taiwan, and others cannot well predict the efficiency and accuracy of the medicines. They have not accounted for the detailed mechanics of the fourth generation and the interactions of different things, which involve very complicated interactions, electric interactions, hydrogen interactions, and chemical interactions, and molecule and for example, the ions and very complicated. Only have you consider this mechanisms in the AI techniques, you can have a high increase of the efficiency and accuracy. Otherwise, you can make a simulation by using the AI techniques, but the result may be wrong. So the PINN have been proposed, but the PIN is still limited to very simple problems. For example, the engineering structure thesis. So that's why we want to develop such techniques.
All right, so we still have several minutes for Q&A. Any questions? Okay, yeah, Professor Wang.
Yeah. You mentioned the cancer cell can pass through confined space. How about a normal cell? If a normal cell can't pass through the confined space, what's the driving force for cancer cell?
Yeah. Yeah, thank you, Professor Wang, for this excellent question. Yes, the normal cells can propagate in the structures, but the cancer cells have some special behaviors. They have some special chemical structure, chemical component, and the chemical behaviors. And they can pass through from our blood vessels and other tissues to enter the blood vessels, and then they will find another position to make a new home. So they have some special molecular thesis, and they have some further. They can make a transport of this molecular thesis by using some vesicles and other things before the propagation of this in the three-dimensional structure thesis. Yeah.
Okay. Yeah.
Thank you, Professor Feng, for your inspiring and informative talk. So because my work includes hemodynamics, so I always feel very interesting about the rhythms inside the human body. So today I learned even for cells, cellular level, there are also oscillations or waves. So I want to seek your comments about whether this level of oscillations has like a relative stable frequency, because like the heartbeat, like the EEG, they all have their own frequency range, right? But how about cellular levels? And if there is a relative stable range, what kind of mechanism can control the cell to oscillate in such a specific frequency? Thank you.
Yeah. Yes, this is in fact a very interesting problems. The cell has a cytoskeleton. It can sense the surroundings. Okay, and on the surfaces of cell and ECM, there are some structures, focal adhesions, podotherms. If you observe these structures at micro scales, they all oscillate. Even the podotherm, they oscillate, and they make oscillation forces to the structures, to the cell skeletons. So the skeletons have oscillation in a periodic of 20 or 30 seconds. So you can observe the rotating or the expanding, shrinking oscillation, different type of the oscillation. Okay, this is important for the cell to move and to tell other functions. For example, if you can imagine how a cell migrate on a flat substrate, why it change is a different ways. That's due to the oscillation. This dynamics is very similar to the movement of a caterpillar, 毛毛虫. Its oscillation is shrink, expand, shrink, expand, shrink, expand. And the oscillation, the rotating oscillation, to change the direction of the movement of the cells. So you can determine how the cells will change its directions in the length scales. I think it's a very, very interesting topic.
Yeah, thank you. Any more questions here? All right. Since we don't have further questions from the online audience, so if no more questions, thank you very much, Professor Feng, for the wonderful talk.
Thank you.
Thank you.
To express our appreciation to Professor Feng and the Purdue Academy for Interdisciplinary Research, prepare this special souvenir for you with your name, titles, etc. in it. Yeah, and then I also wanted to thank all the audience and especially for those who asked questions. And now this lecture is adjourned. Thank you very much.